Okay.
Okay.
All right.
I can not do what I had intended to do for a few minutes, but go back and talk about inhibition a little bit, because Dr. Grisham thinks that some of you might have gotten confused, that I probably didn't explain it well enough last week. But I just want to talk about a little bit for a moment about our remaining agenda. Starting on Monday, those of you who have lab on Monday, you're going to figure out which of the cells of some of these 25 or so are involved in swimming. And we'll talk in a few minutes about how you do that, assuming we have time. If not, we'll explain it to you at the beginning of the lab session. Then, after you've identified all the, you'll identify the cells as a class, and you'll say these, however many, these 10 cells or whatever it is, are involved in swimming. Then, after you've done that, before you go on, I'm going to spend half an hour, 40 minutes with you, talking about an approach that is a little different from what we're going to urge you to use in swimming. I mean, if you, well, I don't know if I should say, I'm probably going to say something wrong. Bill's probably going to, Dr. Grisham's going to kill me for this. So we're going to sort of guide you through it and give you a strategy for solving swimming. There are lots of different ways to solve swimming. Very different ways to solve swimming. In the end, you can come to a pretty good understanding of how it works, coming by many different routes. We're going to give you one that always works. We made it the route that we thought was easiest for everybody to understand it, so everybody hopefully will get it and understand it pretty well at the end. If somebody thinks they're clever enough and wants to go off and do it a different way, totally different way, that's okay, as long as you get, as long as you don't make any mistakes, as long as you do it right. So that's okay. But prior to us starting out on this trek that we're going to encourage you all to use, or at least guide you along to help you, I want to talk to you a little bit about some other approaches. So I'll do that for about 30 minutes or so before you start. And then, after I finish with that, excuse me, you will go to trying to figure out what the interconnections between these cells that you found are, which ones excite and which ones inhibit which others. There will be some issues there. One issue in doing that is that you both have, you can pretty fast come to a pretty good hunch as to what connects with what in various ways. But then you have to do some, as we'll explain either later today or as we'll explain next week, you have to do some experiments to really nail it down, because the hunches can be wrong for various reasons. You can get fooled. We will show you how to nail one. We will get you to nail one, each of you to nail one down during class. But you'll have many more synapses than one. You'll have to nail them all down. You have to basically provide very clear evidence for all of them that you think are there. And most of that, we're going to have you do it. You should do at home. You shouldn't sit in the lab and do it, because you'll spend all your time doing that, and that's actually the easy part once you know how to do it. So it takes some time, but doesn't require very much brain work once you know how. So we're going to urge you to not spend all your time in lab doing a perfect job, but to get on to the basic problem of really trying to figure out how swimming works once you think you know the circuit. And that's what this is, and you'll spend hopefully some of the time this week and certainly the last week doing move on. Okay, so let's go. I need to go back to last time's lecture just for a few minutes. Right. So this, I think some of you got confused by this slide from what Dr. Grisham heard. So basically, when you have excitation, sodium goes into the cells through sodium channels. And when you have GABA release, it opens up to chloride. There's more chloride outside than inside. And the voltages are such that chloride has a tendency to go in under resting conditions, and so a little bit of chloride goes in. Very rapidly. But if the inside of the cell, if the chloride makes the inside of the cell, minus 65 milli. So normally, in this cell, the resting potential is minus 60. If the chloride makes the cell minus 65, or if you make the cell minus 65 by passing some other current into it or something like that, that will be just enough to stop the chloride from going in. That's why we call minus 65 the chloride equilibrium potential. This is all stuff that if you don't remember it from Psych 15, you ought to go back and review it a little bit. If I might as well mention here, to save some possible time later on, if you were to bring the cell membrane to minus 75 or something like that, so the inside was quite negative, and then and then dump GABA on it, what would happen is chloride ions would go out instead of in, because the voltages, the voltages would be such as to push the chloride out, and so your IPSP would be inverted. It would be upside down. You all did the experiment showing that that happens this last week, and that's what was going on. Okay. Now, the issue that I want to talk about right now just for a few minutes is when you have both an excitatory and an inhibitory action going on. There are two different ways to look at it. One is the way that I've shown in black, and one is, actually, that's a bad slide. I can't even see it. I'm sorry about that. I'll have to make it bigger next time. But, oh wait, I can make it bigger by projecting it properly, can't I? In fact, let's do this differently. Okay, that's better. Okay, let's look first at the red explanation. So if a little bit of chloride, of course, comes into the cell, if just the inhibitory terminal is active by itself. But if they're both active, suppose, just for the sake of discussion, which isn't normally the way it happens, but just for the sake of discussion, suppose that the excitatory one got going first. So the cell, no, I'm sorry, let's do it that way. The inhibitory, let's start doing it the right way. Okay, the inhibitory one's active, and some chloride came in and hyperpolarized the cell, but the cell got sufficiently hyperpolarized and no more chloride comes in, so you've got a little IPSP there, hyperpolarizing IPSP. Now we activate the excitatory synapse, and so sodium comes in. That makes the inside of the cell more positive, okay, and that then allows more chloride to come in. And see, what was happening up until that point is you had some chloride come in, the inside had gotten kind of negative, that's keeping any more chloride from coming in. Now the sodium comes in, makes the inside real positive, that just attracts more chloride in. So the chloride's a negative ion. So you've got this negative ion coming in, and that then opposes the effect of the sodium that it came in. So the sodium came in, it depolarized the cell, made the cell more positive. Now that drags in more chloride, and that nullifies some of that positivity. So basically what happens is the EPSP itself shrinks. So two things happen during inhibition. If this was an EPSP in the absence of inhibition, and here's the same EPSP riding on an IPSP, what happens is it will look, well, maybe that's exaggerating, but let's say like that. I don't want it to look like it's right there. Let's take it a little bit bigger still, like something like that. So it starts at a lower level, and that helps cause inhibition, because it starts from farther down, farther away from the critical firing level, which is up here somewhere. But it also, the distance from here to there is less than the distance from here to here. And actually, I really probably should, this is probably in many cases it's more like this. It really wipes it out totally. And that's because, or one way of looking at that's because as soon as the EPSP started, it dragged in more chloride ions, which then nullified it partway. Another way to look at it, same thing, same facts, same physics, same chemistry, but just a different way to do it, is to talk about it the way physicists talk about things, just in terms of current flows, not worry too much about what ions do and what. And one way to think of it is that when if the inhibitory terminal has opened up a big leak in the membrane, so ions can flow back and forth, so electricity can flow back and forth, then when you activate the excitatory synapse and the inside becomes positive, and that positivity tries to spread along the cell membrane to other places to start the spike somewhere else or whatever it's going to do, it leaks out through the hole. It's sort of a short-circuiting action. This hole essentially shorts out, partially shorts out the EPSP. And that is sometimes, that short-circuiting, it's a short circuit, or what a synonym for that in electricity is a shunt. the electricity, or the positivity, gets shunted from going like so, which is what you want it to do, or what you want to do if you're going to get excitation and cause a spike, to going just back across the membrane, sort of like railway cars go onto a shunt off to the side. Okay. I think that's enough. Is that enough of that, Bill? Yeah. Yes.
So essentially, the chloride channels are permeable to sodium.
No, it's not the. No. That's why I said we're going to switch now to a physics explanation. In this last thing, we're going to switch to a physics explanation and talk about just current flow without worrying about what ions are moving. In actual fact, it's not the chloride channel, it's not the sodium channels that move back. Sorry, I think what you probably would have wanted to say, I'm not sure what you did say, but what I think the intent of it was that maybe the sodium ions are moving out the chloride channels, right? And that's not what's happening. The sodium is pushing other ions along and making more positivity there, and some positive ion that can go through the channel goes out, or what really happens is it drags a negative ion in, which comes the same thing. When you start thinking about physics, remember you have your electrons and you have your positive charge and your negative charge. It's really the negative charge that moves, but you don't talk about it that way. You talk about it as though the positive charge moves because it's easier not to keep track of both. So I've said here the positivity moves out. That I'm speaking as a physicist would when I say that. In fact, it's really what's happily happening, since that's a chloride channel, is the negativity is moving in, but it comes to the same thing. Okay? Okay, now let's go back to this time. Well, so in this case here, you think of the order of events. First thing that happens, you get your EPSPs and IPSPs, and then a spike does or doesn't start. And once a spike starts, none of this is going to stop it. It's going to keep going. Is that already in it? No. Okay, now these events reduce the effective amount of excitation. I'd say the inhibition reduces the effective amount of excitation. They basically cause the EPSP to not get as close to the critical firing level as it otherwise would have, both because it starts from lower down and because it goes up less itself. And so if it was big enough to start with, it might just decrease the probability of a spike and not absolutely prevent it. But if it was more borderline to start with, then it actually it will probably absolutely prevent spiking.
Or we might use some more EPSPs to cause that.
More EPSPs might come in and do it, and they would be inhibited too. But maybe inhibition doesn't, it may not be strong enough to stop it. It may just be strong enough to cut down the likelihood of spiking or whatever. There. Okay. All right, so the first thing you're going to do in the lab next week is try to figure out which of swimming's various neurons are involved in swimming. And one good way to work in that direction is to make the following logic. If you've got neurons in the nervous system, is to look at the activity of neurons one at a time, look at them, and if you see that there's a neuron somewhere in the nervous system, an interneuron of some kind, that's firing right in sync or almost in sync with one of the motor neurons, one of swimming's motor neurons, maybe it's firing it at the same time, or maybe it's got a spike right at the beginning of each of one of the motor neurons, the motor neuron burst on one side or the other. So for example, if you had a neuron that fired, these are the motor neurons on the top, fired here, once here, once here, once here, once here, once here, or something like that, you would say, I bet that neuron's got something to do with swimming, right? Indeed, in that case, if that's what you saw, you would probably think that one single spike was triggering a burst or something like that. Or if you have a neuron that basically fires pretty much at the same time when this guy's firing, when this guy, goes brr, 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 more or less in sync with this fellow, you'd say that guy's probably involved in swimming. So you're going to play that game, and you'll find all the neurons that do that, and those are probably all involved in swimming. Now, that doesn't guarantee you to get everybody. It could perfectly well be that you have some neurons, for example, that are maybe spontaneously tonic, that are endogenously tonic, or they're tonic all the time, that feed into the circuit, and without that continual input, the circuit wouldn't be able to do its thing. You could imagine that kind of possibility. I'm telling you as a generosity of my heart that when we designed SWIMMY, we decided not to put any neurons like that in. So that would complicate your life if they were there, but we're just giving you as a gift they're not. But in real life, that's something that would be a possibility. So in this picture, which of these guys, this one, this one, this one, this one, which one do you think is probably involved, which are and which are not involved in swimming? So is 9 involved? According to what I just said, it could be, but I'm telling you it's not, so that doesn't count. 9's tonically active. Is cell 12 involved?
Yes.
Probably. Is cell 11 involved?
Yes.
Think so? It does fire every so often, but I'm not sure it does it in sync with anything or in any fixed time relationship with anything. So it fires here, which is looking like at the very end of that burst. Again, maybe I'm not sure it's firing there. It fires around there, which is kind of the middle there. It fires here, which is so it fires here, which is in the inner burst interval for the left motor neuron. It fires here, which is kind of towards the beginning, if I'm reading upright, of that burst. It fires here, which is kind of the beginning of that burst. Here's a burst which doesn't seem to have anything in relationship to it. So it is firing, but the timing isn't really right for imagining that it's involved in this swimming activity, I would say. You know, if you choose it, you'll play with it, and you'll find out it'll probably drop out in your further analysis, but I wouldn't select that one. What about this guy? Yeah, yes or no? Shout it out. No, right. Yeah, I don't think so. It's not doing any. I mean, it's doing. You have to look at it, you know, carefully, but I don't think it's doing anything in any time. Yeah, that's synced to either of these two fellows. So on this picture, probably only that one would be a good candidate, and there'll be some more. So you'll go through that procedure at the beginning of the lab, or you can do it at home if you have Swimmy downloaded. No, sorry. Okay, that's okay. Now, once I have finished my once you found all the cells and I finished my little spiel, my 30 or 40-minute spiel about another approach different from the one you're going to use. First of all, we've talked about, okay, 1 and 2 are the motor neurons. You already know who they are. They're 1 and 2. You also know from our discussion of last week that the reason one seems to fire, at least in part, is because it gets input from somebody else. Remember we saw the EPSPs in it, so those EPSPs had to come from somewhere. So and what's true of 1 is true of 2 because this circuit is bilaterally symmetrical. So both 1 and 2 have 1 or more other neurons that excite them. So the first thing you're going to want to do, or one thing that's a very plausible thing to do first in trying to work out your circuit is to find which ones those neurons are. Another thing we learned is, remember there was that reciprocal inhibition which worked via an inhibitory interneuron. So another thing you might do is look and actually find the inhibitory interneurons. You know they're there, so find them. Okay, and that will get you started. Then that's not all of them. There are going to be some more, but those would be good ones to do first. And what we'll do next during the lab, what you'll do during the lab is you'll sit there each independently working on this, and when you find one, you'll raise your hand, and the TAs will come over and see if you convince them that you got it right. They'll announce it, and then we'll put it up on the board, and so people can stop. You know, people will then know about that one. So you don't all have to find every connection. You'll work as a class to work out all the connections because it gets kind of tedious otherwise. And because, of course, science fundamentally is teamwork, and so that makes this a little bit more like real science. One person can never do it all. Now, what's this? I have no idea what this is. Oh, this is looking. Yeah, let's not bother with that. This is looking for the inhibitor. I don't want. Oh, there is one thing I want to point out, though. Actually, maybe no. We should do this. This is not looking for the inhibitor. Let's see. Looking for a cell that directly excites cell 1. Okay. So here's cell 1. That's one of our motor neurons. And if you right. No, I do need to do this. Here are some other cells. Okay, that are in fact part of the rhythm. I have removed any labels that will allow you to identify them, so it's not going to help you. But the question that you're going to be faced with is, given a picture like this, suppose you're looking for whatever it is that produces the EPSPs in cell 1. Remember, that's the first thing you're going to look for. Well, it is one of these guys, and the question is which one? Well, it's clearly not that one. 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 It could conceivably be, just glancing at it, that one, or that one. And then the question, which one is it? First thing to remember is what you're looking for is a cell that spikes, and then and then 1 millisecond later produces not necessarily a spike in its direct postsynaptic target, but produces a synaptic potential. It's the synaptic potentials that are delayed by 1 millisecond, not the spikes. That's the synaptic delay. And so what you want is a cell that, and there's an obvious EPSP there, but remember we discussed the fact that each of these spikes is sort of emerging from an EPSP. So what you want is a cell that spikes just a little bit before here and here and, here and there and, what's that? Oh, I see, that's a late spike from that fellow. Well, actually, what is going on there? Oh, yeah, I guess and there. Okay, so let's see. Is there anybody? Well, so this fellow here. This one, look, it's got spikes way before, and then it's got another one that's kind of synchronous with that. That's not a good candidate. But this guy, it's got a spike here, and there's an EPSP and then a spike. It's got a spike here, and there's an EPSP and no spike. It's got a spike here, which I didn't draw a red line, which is what confused me. But that is correlated with that EPSP and that spike there, and so on down the line. And so a little bit after each of these guys, there's an EPSP. And in order to show that it's really a 1 millisecond synaptic delay, you can't really do it at this time scale. You want to spread it out side to side so that you can really measure the time accurately. All right, I guess that's all for now.